originally from Kiel in Germany, but he's working for the Scottish Rural College that is in the same building of the Roseley Institute, I guess, is it? And he's going to tell us about the importance of microbiome, and metagenetics in production, animal production. This is a top subject, and I remember that we were, where is Miguel? We were in Denmark. He was presenting his studies on genomics applied to feed the fishes in cattle, and somebody in the audience raised their hands. Did you study the metagenomics of these animals? And he said, no. So, okay. I'm not interested in your results. Metagenomics probably is more important than what you did. That really impressed me, and that's one of the reasons we brought Heiner to talk about. Please, thank you.
Okay, thank you very much. Thank you very much also for the invitation, Bento and Eiji. It's a great pleasure to be here and to talk about the subject where I'm most like to talk about. It's the microbiome. It's so interesting, it's so informative, and I would like to give you an overview about this informativeness. And this time I will talk not alone about the animal influence, I will talk also about plants, to give really a big overview how you can use the microbiome, how you can use the systemic effect or symbiotic effects of the microbiome. Now this one is... First of all, I want to define the microbiome. The microbiome is a collective genome of residence microorganism in a host. And we as humans, we have also a lot of microbiomes in our gut. It's about 100 trillion of microbes, and they are representing 2 million of microbial genes. So 10 times more microbial genes you have in your gut than the home genome has. Now is the question, is it important also economically, the microbiome? And in human, the microbiome or the human microbiome market is in 2017 about 260 million, and it increases to 1.2 million probably in 2025. And in agriculture, it's already a big market. In 2007, already 3.9 billion, and the expectation is that it increases to 6 billion in 2022. Now the microbiome I want to get first from the soil to a human, and the soil is a very interesting kind of subject, because if you take a teaspoon of soil, in this teaspoon of soil are more microbes than humans are living worldwide. So it's a very big ecosystem, and this ecosystem converts, for example, organic mass into nutrients the plants can absorb, and plants also have microbes which are penetrating into the plants and stimulating growth. And of course, animals have a lot of microbiome in their gut, and this microbiome has an influence on welfare. Humans, and I have always to emphasize that you have to feed your microbiome right in order to be healthy, and there are now a lot of studies showing how to feed your microbiome in order to be healthy. What is now the microbiome? The microbiome is the bacteria, the viruses, the fungi, the archaea, the nematodes, and protozoa, and they are all symbiotic with the animal, with plants, and there are only a few pathogenic. So the symbiotic effect is overwhelming in nature. Now coming to the soil ecosystem, the microbiome decompose organic matter towards humus, fixation of nitrogen and other nutrients, protects the roots from diseases and parasites, provides nutrients to plants and produces hormones to stimulate those plants, improve the structure of soil particles, and alleviate it from stress like drought and other stress factors. So this already shows how important the microbiome in the soil is for the plants. And plant growth is depending highly on the microbiome. Of course, it's also influenced by climate, by soil structure, soil nutritional quality, and pH, but it's always a symbiotic effect with the microbiome. And this kind of symbiotic effect is very well known in legumes. There are rhizobia, which is located close to the roots of plants in so-called nodules, and they are fixating atmospheric nitrogen into the plants. They're converting it in nitrate, which can be absorbed by the plants. But it's a symbiotic effect. So the plants providing in exchange carbon in order that these microbes can work. And over the whole kind of presentation, I wanted always to show that it's a highly symbiotic system. For example, in plants, they are influenced by a lot of stressors like drought, nutrient deficiency, diseases, parasitic weeds, salinity, and the microbiome can alleviate these stress factors. And that's also where the business are working. They are producing substance in order to provide a microbiome to reduce the stress factors for the plants in order that they can also grow under very harsh conditions. And here is it shows the industry, what they are providing in plant microbiome, the biofertility inoculations. That means to improve nitrogen fixation in the soil by microbes or to make phosphate more soluble by microbes so that the plants can absorb phosphate. But there are also biocontrol microorganisms which are working on strategies of antibiosis in order to remove pathogens, competing about space with pathogens, working on enzymatic activities, or inducing even resistance in the plants. That is a big business in plant management, plant improvement of the environment, but there's also coming up a big business in animals on the microbiome. And in animals, one trait which is very important is feed conversion efficiency. And we are working on bovine and on ruminants, and there the rumen is a very interesting ecosystem of microbes which are converting fibrous food into different nutrients the animal can absorb. And improvement of high feed efficiency is of high economic impact, because 60 to 70% of the variable costs are feed cost, and feed is a limited resource, and we should use it as efficient as possible. But there's one disadvantage for the environment on this system of fermentation of food in the rumen, that is methane production, and 40% of the total greenhouse gas emissions in livestock is due to methane, and therefore we are always blamed in beef production that we are producing too much methane. But we want to develop a system where we not only improve feed efficiency, we also want at the same time to reduce methane emission by breeding. And therefore we are taking rumen samples in order to see whether the rumen microbial community can be used to predict feed efficiency, and then to improve feed efficiency by the predicted feed efficiency, for example, with genomic selection. But for this kind of system, we need a good selection criteria, and we are in the process to provide this selection criteria, and I will show you later on what kind of selection criteria we are recommending. It is not an easy task to disentangle the influence of diet, host genetics, and rumen microbial composition. And therefore, we have to do very detailed experiments to disentangle all these effects. And in these experiments, we are measuring feed intake and then calculating feed efficiency, and we are measuring also methane emissions. In ruminants, the microbiome converts fibrous food into absorbable nutrients which are of high quality. And the interesting part is the density of the microbial community in beef cattle. For example, in 1 gram of a digester are 10 billions of microbes. In 1 gram of a digester are millions of protozoa. There are thousands of fungi. So it's a very dense ecosystem. And these microbes are producing, due to fermentation, volatile fatty acid, which the animal can use for energy. They are producing microbial protein, which the animal digests in the abomasum and then use as protein sources, and it produces B vitamins. But the disadvantage, as I said earlier, the animals are producing methane, and 95% of the methane are belched out of in the atmosphere, and therefore we want to reduce these methane emissions. Methane emissions is based on an excess of hydrogen produced by bacteria, protozoa, and fungi, and methane is produced by methanogenic archaea. And therefore, we are looking at archaea, bacteria, mainly in our further studies. But to do everything, we need to record feed intake, and we need to record methane emissions. And we have a very nice facility with 6 methane chambers where we individually measuring methane on an animal, and this has been shown that it is very accurate, and we get very good results out of this methane chamber. But in addition, you have to always record also feed intake, because both traits are important. Now, we were interested in whether there's a host genetic effect on the microbiome. And we made the trial in that way that we could predict the sire effects. So we have taken sire progeny groups. So we tested on average 8 progenies of each sire. And then we looked at these estimates of the sire effect. And we found significant differences of the archaea bacteria ratio, of the sire effect, so that would indicate that there's a host genetic effect on the microbial community. Then we looked on the same data, the sire progeny groups on methane emissions. And we found also significant differences in sire effects, so genetic effects in methane emissions. And the striking point on this analysis, the ranking based on the archaea bacteria ratio of the sires is the same than based on methane emissions. So that means the host genetics change the microbial community, and a change of these archaea bacteria ratio resulted in a change of methane emission. And that's the fundamental point we wanted to test, whether there's a host genetic effect on the microbiome. If there would be no host genetic effect, then we could not use this kind of information in breeding. But this host genetic effect has not only shown by us, it has been shown in different species like mice, humans, pigs, dairy, sheep. And this is also a nice study by Weimar. They exchanged the rumen content between cows and after 2 weeks, the rumen content of these cows changed back to the original rumen content. And that's also an indicator that the host genetic must have an influence, otherwise they would stay with the rumen content they have got from the other animal. But it is always important not to look only on statistics. You have also to look on the biology to see whether from the biology point of view also your results make sense. And why are now a host genetic effect on the microbiome? One important effect to determine the microbial community is the pH. And the pH depends on saliva production, because in saliva is bicarbonate, and it regulates the pH in the rumen. And there's a large variation of saliva production. And cow produces 150 liters of saliva per day. And 30 liter variation is found between these animals. So there's large variation available in saliva production. And it is possible, and we assume, that saliva production is also determined by genetics, and therefore the saliva production is one indication that the host genetic effect has an influence on the microbial community in the rumen. Of course, differences in short-chain fatty acid absorption has an influence on the pH and then also on the rumen microbial community, the passage rate of proteins. And the passage rate of proteins in the rumen is also depending on the physical size and the structure of the rumen, and structure and physical size of a rumen has, of course, also a genetic component. Very important, where a lot of people are now working on, is the passage rate of digestion, which has been shown to have a big effect on the microbial community. And where people now mainly working on is this microbiome gut-brain axis, so that stress of animals has an influence on the rumen microbial community. The immune system has an influence. And later on, I will show you some results where we are looking at Fucose sensing and its influence on the microbial community. We call it even crosstalk between the microbes and the host animal. And here is shown how it is more and more explained, this interaction between the microbes and the host. As the microbes, they are producing metabolites which are going over the bloodstream and have an influence on the brain. The microbes have an influence on the immune system. The microbes have an influence of the neural pathway and influence, and then the brain. But it is not one way from the microbes to the brain. It's the other way around, over the HPA axis, over the endocrine pathways. The host influences the microbiome. The immune system of the host influences the microbiome, and also neural pathways from the brain have an influence on the microbiome. Now, we are working on a total new ball game because of the advance of sequence technology. we can sequence the whole metagenomic community. And that's what we are now doing mainly, that we are taking rumen samples and sequencing the whole microbial community of this microbiome. And then we can build on that information, decide which kind of microbial community is available. You can determine it on different levels. At the highest level, it would be domain, as we did at the beginning in archaea bacteria ratio. At lower level, it's on genus level. There you get more specific kind of archaea, in this case, Methanobrevibacter, Methanosphaera. But what's striking on metagenomics is you get also the abundances of the microbial genes. And these microbial genes, we know really the function of these microbial genes. And that is a kind of information which is unique and therefore so powerful. Here we have identified about 4,000 microbial genes, and then we did a network analysis of these microbial genes. Every dot is a microbial gene, and this network is built only based on the abundances of these microbial genes. So if they are very close together, then they are correlated, these microbial genes, and their abundance is then correlating. So they have a kind of functional equal regulation. And then we looked at that, and we found in these two clusters, 4 and 6, were mainly genes which are associated with methane emissions. So we know the genes which are in the pathway of methane emissions, and we were surprised that those are mainly in just 2 clusters. And these genes, we wanted then also to see what are the most important genes of those. And we identified 20 genes which could estimate the methane emissions by an explained variation in methane emission of above 80%. And that's quite a striking result. Of course, you have always to consider we did this analysis based on animals we selected for high methane emission and low methane emissions. And here are all the different genes which are associated with methane emissions we found. And here it's ordered the animals by increase in methane emissions, and that's a heat map. Blue indicates a low abundance of these genes, and yellow high abundance of these genes. And you can see with increase in methane emissions, all these genes increase in abundance. And then you look in the pathway of methane emissions, and here in red are all the genes we identified to be significant with methane production. And you can see that they are all triggering specific kind of metabolism. And that's very informative because you're estimating something which has also a functional association. And then we looked into different diet effects on methane emissions. And it is known a specific kind of gene, the mcrA gene. It is catabolizing the final step of methane production, not only in cattle, also in digester and you see here it increased the relative abundance, and then methane emissions also increases. Of course, it's different for different diets. We know concentrate diet has a lower amount of methane emissions than a forage-based diet. But if you see the slope is quite equal, so it's a more kind of constant effect of concentrate and if we can consider, for example, in breeding the diet effect, then we can use this kind of information easily to predict, for example, methane emissions, or to use this information in genomic breeding. We did further studies, and we validate everything from the first study where we had lower amount of animals, in a bigger study where we had more animals. The microbial community provided additional information to, in addition to the microbial genes. But microbial genes are most informative, and we found less diversity of the microbiome in low emitting animals. Now we are looking always also in this network because I think they are very informative. The dark matter, that are all the microbial genes, and then we are overlapping the microbial community over it. And here is one cluster which is mainly associated with Methanobrevibacter, and there are a lot of genes also involved in methane production in this. And we get also other kind of microbes identified, and that's quite interesting because then you can see what association is between the microbial community and the microbial genes. And based on the microbial genes, because we know their function, we can also then identify what these microbes are involved in what kind of metabolism these microbes are involved in. And for example, the cluster with Methanobrevibacter showed, of course, higher methane emissions. Other clusters, for example, Proteobacteria, Selenomonas, they showed low methane emissions. Then I'm coming back now to feed efficiency, because we were not only interested in methane emission, also in feed efficiency. And here's the same kind of network. Here I showed you the different methane emission genes are in these two clusters, and for feed efficiency they were in totally different clusters, in 2 and 5. And that shows on the microbial level, it seems that feed efficiency is independent from methane emission. And we identified 49 microbial genes, and they explained, again, 80% of feed efficiency. And a lot of these genes associated with feed efficiency have also a biological reason which we can relate to metabolism of feed and to metabolism of which is necessary in growth. But I will concentrate on two quite interesting genes. Fucose isomerase and GDL Fucose synthase. Fucose is a component of the innate immunity. It is within the mucin layer of the rumen wall. It is produced by the intestinal mucosa. It is produced by saliva, and it keeps the integrity of the mucosal barrier. And bacteria, they demand now for fucose because they need fucose to grow. And bacteria have a specific kind of factor, it's called FUCR, which initiated that the host produced fucose for the different microbes. So the bacteria are signaling the host to produce mucin. And the control depends, of course, on the demand of the bacteria. But why is now the host producing mucin or more fucose for bacteria? The host produced it mainly to make the mucin layer thicker, to keep out pathogens. And on the other side, the host also produces it mainly to grow commensal kind of bacteria, which are symbiotic with the animal. So that's these kind of crosstalk between the microbiome and host. Here is again, it's shown how we think the fucose protects against pathogens, and if there's not enough fucose, these pathogens can penetrate into the intestinal mucosa and then harm the animal. But there are also other factors we found now. We found in the high efficient animals also more genes which are, we found in the low efficient animals more genes which are producing biofilm. And biofilm is the protector of pathogens in order not to be tackled by the immune system. And the opposite we found in high feed conversion efficient animal. There not so much this microbial gene to produce biofilm has been found. So there's another reason why feed efficiency is controlled by the microbiome, and that there's a crosstalk between the microbiome and the host. Now we are looking not only on feed efficiency, because the trait which is most difficult to measure, to calculate, is feed intake. So we are looking at feed intake, and we did that in groups. We selected for a low feed efficiency and selected for high feed conversion ratio. And you see there's quite a lot of difference between these groups. So there's also a high variation between animals, which indicates that we can also select for feed efficiency. Now we wanted to predict daily feed intake. And we found that 23 microbial genes can predict daily feed intake by 70% of its variation. So again, daily feed intake can be predicted by the microbial genes, and therefore used to predict those traits. For other traits, we needed more genes to identify for feed conversion ratio in this data set because it is a more difficult trait because it's a ratio of 2 biological rate. Interestingly is also what kind of genes we have identified. For a daily feed intake and residual feed intake, we identified 9 genes they have in common, and the other genes are for each of these traits. For average daily gain and feed conversion ratio, we found 80 genes which these 2 traits have in common. But we did not find any kind of genes between average daily gain, between the group of average daily gain and feed conversion ratio and those genes associated with daily feed intake. So that's again showing that these microbial expression has a different influence on different traits. Now we looked again on these networks and network clusters. So we found one network cluster which was only influencing feed conversion ratio and average daily gain, other network cluster which only influence daily feed intake, this cluster only average daily gain, and one cluster was associated with all traits. But this is quite important. So we're finding different genes to use to predict different traits. If we would find always the same kind of traits for all the different information, or in this case, different daily feed intake and feed conversion, then we could not disentangle all these different traits. Also, we are looking now at the function of these different genes. Microbial genes, here is a hub gene which influences highly daily feed intake, and it is associated with starch and sucrose metabolism. There are other genes which are mainly associated with galactose metabolism, with lactate transport, which are influencing feed conversion ratio. We are looking also at average daily gain. There again, other kind of hub genes are associated with average daily gain. So we can really distinguish between these different traits based on different microbial hub genes. Now, from breeding point of view, we want to use this kind of information. We can take rumen samples on live animals. We can take rumen samples in the abattoir. Then we have to determine the abundances of these microbial genes I have shown you before. And then based on the abundances of these microbial genes, we can predict methane emissions. We identified 20 genes which are associated with methane emissions. The 49 genes I showed you can be used to predict feed efficiency. Of course, the microbiome has a big influence also on health. So we are also interested to predict from the rumen microbiome health traits. And then we can use this information to predict EBVs or GEBVs, genomic EBVs, for this different. The advantage is really we don't have to measure these animals based on daily feed intake, based on methane. We are predicting from the microbial gene abundance those traits. And we can take a rumen sample in the abattoir and then predict from these rumen samples the animals in daily feed intake, in average daily gain, and other traits. And that's so interesting for these, for breeding purposes. That means this has an advantage for breeding companies in order to save costs in measuring traits. It is more cost-effective, and the microbial genes are showing high metabolic background. And I said at the beginning I will identify the best selection criteria, and the best selection criteria is rumen microbial gene abundance, and this is a new phenotype we can put into breeding in order to reduce methane emission and to improve production efficiency. We can use this abundance of the microbial genes also for other traits. So if we are taking a rumen sample and determine the microbial gene abundance, then we can relate it also to other traits. Of course, other microbial genes will have then an influence on animal health and animal behavior, and even meat quality. We are looking at fatty acid profiles in meat and associating it to the microbial community. And I started with plant breeding, and the same can also be done in plant breeding, of course, to identify all the microbes in the plant or next to the roots of the plant, determine the abundances of these microbial genes, determine the performance of the plants based on genes, predicting the nutrition efficiency of those plants, and also the resilience or resistance of those plants to stresses. We also using the microbial community information to identify antimicrobial resistance. So it's another byproduct what you get out of the rumen microbial community. We identified 204 microbial genes which are associated with antimicrobial resistance, and that's a big topic in Europe to reduce antimicrobial resistance, and we get that automatically with our samples as well. And here shown the antimicrobial resistance genes and the pathogenity genes we identify. And you see it has a totally different abundances in the forage fat animals than those animals we offered concentrate. So there you see that the microbial community also in the antimicrobial resistance is totally different depending on diet, and the diet mainly based on concentrate showed a lot of antimicrobial resistance genes and showed also a high protein, high proteobacteria to firmicute bacteroides ratio, and this kind of ratio is used in human to identify healthy guts and unhealthy guts. So this indicates also that the concentrated diet, those animals, even though they did not show any kind of diseases or health impairment, they still have not an optimum microbiome. We call it is a dysbiosis in the microbiome. So the microbiome you can use for plant, for animal, but you can also use the microbiome for high quality food. You can use the microbiome to work on manure and waste management. The microbiome is big in biodigester, so there are so many different opportunities to use it. I will only mention the food production, and I like very much wine, and if we can even improve the wine by the microbiome, so I will be involved in that kind of study as well, and will be the first one to try it. One disadvantage at present is that we do not know very well the microbial communities. And so only a few in the rumen, there have been 400 microbes cultured. And these 400 microbes have then sequenced, and then their sequence have been identified. And they achieved that over 10 years, so it took a long time to get the whole sequence of those microbes. But we are using metagenomics, and based on this metagenomics, we have the whole kind of reads of these microbes sequence. And now there are new techniques of binning to build up the sequence based on metagenomics information. And we build up, from our metagenomics information, 900 microbial genomes. These microbial genomes have never been sequenced before. And we achieved it in 1 year. And we had not so much money involved. So this kind of methodology is unique and gives us very new information and a lot of more research we can do then on our samples because we know more genomes of these microbes. In addition, we identified 69,000 proteins involved in carbohydrate metabolism, and that has never been identified before. So there's a lot of information. Of course, you can also use the microbiome, including gene editing, and gene editing, mainly the the CRISPR-Cas9 system has been developed based on Escherichia coli in order to change it. So, of course, you can also use this kind of technology today. Now I want to summarize that the microbial information can be used for precision in soil management, for plant breeding, nutrition, animal breeding, nutrition management, improvement of waste management, control and improvement of fermentation and food production, biodigester, for microbial genes as biomarker, for example, biomarker to identify stress, to identify health, microbial products for soil and plant. And you can also use the microbial itself to directly produce food. There's another kind of opportunity. Here is in specific for the microbiome in animal production. I showed you methane emission, meat quality, stress. Health is a big factor where you can use the microbiome. Pharmacological interesting gene you can identify in the microbiome. Antimicrobial resistance, resilience and robustness of animals we are after. Probably can identify based on the microbiome the resilience of the reproduction. I did not tackle that. Reproduction has, of course, an influence. The microbiome has an influence on reproduction, identification of malevolence. And now I want to finish with this one, that the microbiome can contribute to alleviate hunger, and by increasing mainly the sustainability of intensification of food production and welfare, not only of the animals, also of humans itself. I want to acknowledge at the end our funders, the Scottish Government, BBSRC, and we had aligned funding from DEFRA, EPLIC, and QMS. And of course, we are working closely together with the Roslin Institute and the Rowett Institute. And the Roslin Institute doing the kind of bioinformatics on the microbial genes we have generated. Is the group around Mick Watson, the information about microbes, mainly in humans, we get from Alan Walker's group at the Rowett Institute, and of course my colleagues where I am working with, they are doing a great job that I can travel here to Brazil and to provide this kind of talk. Thank you very much. Yeah. 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 Okay, that's great.
Now we will have the lecture of Professor Paulo Arruda. You want to introduce him? No? Yes. We will. Nós vamos fazer uma mesa redonda ali com os dois.
Como é que for? Ah, volta aqui, né? Ixi, voltar no menor. Okay. So, first of all, I would like to thank you very much for Zé Bento for the invitation to come to this fantastic meeting. And I'm afraid that you guys is going to have an overdose of microbiome this afternoon, because yes, I will talk a little bit about this also. And well, I think that you already had a presentation by morning today talking about on the problem of sustainability in food production to tackle the problem of world hunger. Just to remind, I think that, sorry if you have had this kind of numbers by morning, but the dilemma of food production is that we need to produce in the next 50 years something like what we have done in the last 10,000 years. So if you take this range and just to compare the size of this challenge, and about 925 million people are hungry. 70% of water is used for food production in agriculture. 40% of the world employment is in agriculture if you take the labor in the underdeveloped country or developing country. 30% of greenhouse emission comes from agriculture. And we need to increase 20%. 20% in production, 20% reduction of gas emission, 20% reduction in rural poverty. And what is the challenge? The challenge is to produce more with less. Less what? Less fertilizer, less pesticides, and less water. Most of the energy consumed by man come directly or indirectly from carbohydrates produced by cereals, vegetables, and fruits. fat from leguminous plants, and protein coming from milk, meat, egg, and fish. But this also depends on plants. So carbohydrate, fat, milk, egg, and meat make up 80 to 90% of energy consumed by man and are directly or indirectly derived from 5 cultures: rice, maize, wheat, soybean, sugar cane, of course, because it contributes a lot with sugar. And to meet the needs for the population, increased population in the world by 2050, we need to increase production by 60%. But how are we going to do this in an environment that is always challenged by climate change events that impact crop production dramatically? This is what happened in the agricultural year of 2015-2016 in Brazil. You see here when you have dark red means that is very drought stress. And if you go from pink to dark red, you see the size of the areas that were affected by drought in 3 years ago. And this climate event decreased the production by about 18 million tons of corn. This is 20% of the harvest in Brazil, right? If you look for this number in US in 2012, you will see that the climate change events decreased the production by 45 million tons. That is about the size at that time of the production of corn in Brazil. And this, of course, has a lot of impact in terms of economic loss. In this year, this crop year, we lose 11 billion reais, about $3 billion only in corn. How can we face this problem? We need to invest in developing crops that are more resilient and then are more tolerant to climate change events. To tackle this problem, we created a center in University of Campinas that is a joint research center between Embrapa and our university, Unicamp. We call this Gene Clima because we are looking for genes to solve the problem of climate change. And this is an Embrapa and Unicamp joint research center. And then when we created the center some years ago, we wrote a very nice story, and we went to the money from Minister of Science and Technology, from the banks, from the industry, from a number of people, and they said, this is a fantastic project. But they did not sign any check. So last year, we had approved by FAPESP this, the money to run this Genomics for Climate Change Research Center. And it was very nice because it's a long-term research program. We got funding from FAPESP for 10 years. And of course, this is reviewed every 3 years. But this is long-term funding is absolutely essential for this kind of work. And what we are running is a kind of a platform that we call From Gene to Trait, and where we have a program for gene discovery, proof of concept breeding, and we want to go up to the pre-launch and launch together with the private sector varieties that are produced in this research program. And one of the most important parts of this work is the gene discovery that use structural and functional genomics and gene editing, gene walls, and everything you can think using genomics. But one thing that is going fantastic well is microbiome. Thank you for introducing the topic for the audience. And after, identifying genes or genes coming from plants and from microbes or for other sources, we make gene construct, we do transformation and phenotyping and so on and so on. So we have built a robust research pipeline of gene discovery and proof of concept that allow the development of proprietary technology. So we need to find something that is absolutely new. We don't want to do what many people do, this more of the same. So we want to find really microbes and genes that have never been touched by any other in the world. So, and this involves a lot of work on the bioinformatics and gene discovery, and we take care also on intellectual property and regulatory affairs, and all of these, of course, we use a lot of information technology. One of the most important programs in the lab is the mitochondrial metabolism genes for drought tolerance that is involved in some kind of mitochondrial metabolism, because mitochondrial metabolism is very important in a key stage of plant development that is very sensitive to drought stress, that is flowering time. Flowering, during flowering, the cells that originate the pollen grain increase about 100-fold the number of mitochondria in the cells. And anything that is change or affected mitochondrial metabolism at this stage causes decrease in production of pollen, and then this affects the yield. So we are interested in a protein that is called mitochondrial uncoupled protein that was discovered in our lab many years ago, and now we know that this is a protein that is very important controlling hypoxia response in plants, and we have done a lot of work on this and using all the tools from genomics, and we have seen that this affect the recovery from drought stress when you overexpress UCP1 is much faster than the control, and this affect the percentage of silics in the plants, and this helps the relative area of seed and seed per pod, and so this is related to seed production. We are also investigating genes from drought tolerance from estromophile plants. So this is an example from a plant that grows in this area in Turkey. This is a very high saline area, and we have done a lot of work on this and select the genes and put the genes in sugarcane and in corn. And you see here, this is control, 9 days drought, stress, non-irrigated. And you see here, control, irrigated, and non-irrigated. You see the effect of drought stress, and this is the gene that we call unknown, because it's absolutely unknown. There's nothing in the literature, and this is something that we like very much, because you can discover many things related to the gene, and this is also introduced in corn. We are also investigating this area that is called Campos Rupestres, that is located in the state of Minas Gerais, in Serra da Canastra, Serra do Cipó. And well, those are plants that are two species, Velosia. One is resurrection plant. This is a plant with 4 months without raining. It dries, completely dry, and 1 week after the first rain, you see that the dried leaves comes green again, and this is a kind of phenomena that we are interested. And this is a very close related plant that never dies. So it's called evergreen. So we are looking for genomics and microbiomics of this area, because this area is very dry. the soil is very poor with very low concentration of phosphorus and nitrogen. But one thing that we would like to explore today with you is how can we plant microbiome health facing drought stress. There's a lot of publications in the literature showing that some microbes that have been isolated and studied by many different groups around the world for decades, and showing that, well, microbes can help nitrogen fixation and phytohormone production and stress and so and so. And so we were asked to do something completely different. So we started this program asking the question, can we map the microbiome of the entire plant? So can we discover the microbiome that lives in the roots, in the stalks, in the leaves, that lives inside the stalk, inside the roots, in the rhizosphere, in the exophytic part of the stalks, and so and so. But when we devised this project, we saw that we want to map using microbial DNA sequencing, using 16S and ITS for bacteria and fungi. This is to have an idea of the diversity and distribution of microbes in the plant, so that we could describe the diversity and abundance of the microbes in the plant, and also to construct a culture collection from this plant so that we can cross-reference the microbe with the data on the diversity and abundance, so that we can take information from this to define microorganism consortia for making inoculum consortia. This is what we submit to a funding partner, and they said, okay, it's fine. So this was done by Rafael Soares as a PhD, now a postdoc in the lab. To do this, we grow a plant in an area. We use plant growing in an area in soil that has never been sprayed with pesticides or have never been fertilized with chemical fertilizers. Why? Because when we show this data to scientific community interested in the area, they say, well, this is not good because you need to go in the field plantation. You need to find the microbiome in the plantation to see the influence of the different varieties and the soil and temperature. But they forgot one thing that is very important. What people use to grow sugarcane, what the sugarcane mills does. They used to fertilize the soils with the fermentation, the most from the fermentation. And what people put in the most to control microbial growth? They put antibiotics. So they spray a lot of antibiotics in the field. And what people spray to control the weeds? They spray Roundup. They spray herbicide. Okay. So perhaps the microbe communities that are in the plantation, the commercial plantation, have been selected for antibiotic resistance, herbicide resistance, and many other characteristics. So perhaps looking in a naive soil could bring something like what have happened in the evolution of this association between microbes and plants. So to do this, we grow the plant and we harvest plants during the different growth stage from 4 months to maturity, and we harvest different parts of the plant, roots, leaves, stalks, different parts of the stalk, and we did this, and then we selected the microbial communities, we isolate the microbial communities from the internal part of the organs and the external part of the organs. So we use this commercial sugarcane variety that is used in Brazil. And also we, sorry. Okay, and we collect also samples of soil around this area so that we can compare what is in the soil and what is in the plant. And we discovered about 23,000 bacteria and about 11,000 bacteria in the 11,000 fungi in the different parts of the plant. So a significant proportion of microbial diversity is shared among tissue, among organs. But you have different populations on different communities when you compare the different organs. And you see here, for example, that bulk soil, root, and young leaves is the most diverse, but you see here that endophytic stalks and leaves is also very highly diverse, but endophytic stalks and leaves, internal part of these organs, the diversity is lower. Plant organs selected their most preferable microbes probably through functional enrichment. Soil serves as a reservoir of for the assemblage of plant organ microbiota. And this is shown here. When you compare what we have in soil with the different part of the plant, you see that, for example, you see here there is a faint red line here and in the soil, and you see here that the red goes up depending on the organ that was sampled. And this is the same this is true for bacteria and for fungi. What this means that when a plant sits in the soil, the diversity that is in the soil is selected by the different organs on the plants along the development so that tissues or organs increase the abundance of the community of microbes, of the microorganisms that perhaps has a functional association with that particular tissue. So microbial groups are either depleted or enriched from soil to the plant organs. All right? So this means that a concept that the plants depends on the diversity that is sitting in that particular soil, right? To do this, to do this assemblage of the microbial communities. But most important, there is a huge diversity, but there is a concept of a core microbiome. What's the core microbiome? Here in this white triangle, you see that about 10 or 15% of all the diversity represent 90% of the abundancy. And this is true for bacteria and for fungi. And the pattern, the core microbiome, we call this core microbiome because this core microbiome don't change too much along plant development. So it's constant, it's highly abundant, and the idea is that those microbial groups that are enriched in plant organs, maybe they are enriched because they may play an important function for that particular tissue or for that particular organ. So this is the core microbiome, the microbiome that represents the highest abundance in a particular tissue and a particular organ. And then we made this culture collection from the same plant, from roots, endophytic, rhizosphere, and stalk. And to do this, we developed a new concept that is called community-based culture collection that we call CBC, because normally what people do is a common conventional microbiology method for isolation of microbes. So first plating series, and then isolate a colony, and again strike in other plates, and so up to pure colonies. And we don't do this. What we do is just first plating, primary plating, and then collect the colonies and store, and that's it. And what happens is that in each well of your plate, you can have 1, 2, 3, 4, 5, 10 microbes. Doesn't matter, because we can identify using PacBio sequencing. Using PacBio sequence, we can annotate completely the collection, bypassing the laborious and time-consuming steps of conventional microbial, reduce the cost and minimize the loss of biological information, because perhaps some of the bacteria can grow only in association with other bacteria, because one species can produce something that is good for the growth of the other bacteria. And all of this, we call it this has been published, we call this community-based culture collection. And then we asked, and this is the entire annotation of the collection. We identify over 500 unique bacteria. Most of those bacteria have been are new, and very few have been described in literature. And the wells of the plates can have 1 to over 7 bacteria in each plate, and so and so. And this collection represent bacteria that you can find in different tissues, in different organs of the sugarcane, including rhizosphere, roots, young shoot, bottom stalk, medium stalk, upper stalk, leaves, and so and so. That means that we have a sample of bacteria that represent the communities that colonize the sugarcane plant. Next question we ask, can we identify high abundant bacteria in our culture collection? And we did this by cross-referencing the culture collection with the sugarcane microbiome map. And this allowed us to identify what we call robust colonizers. What is robust colonizers? Are members of the core microbiome that's very highly abundant in any tissue or any organ of the plant, in the roots, in the stalks, and the leaves, right? So after this, we asked, are those robust colonizers playing something benefit to the plant? To do this, we use corn. For a number of reasons. We select the 10 most abundant bacteria from the roots and the 10 most abundant bacteria from the stalk, independent of the name of bacteria. This is a blind exercise in terms of the species of bacteria. We just use the abundancy, and then we inoculate. We made this, we construct a synthetic community assembled with a set of most abundant bacteria in roots and stalks, and then we put this in corn. And what happens, you see here and you see here, you can have an impact of over 3-fold increase in biomass in this young seedling, of course, young plants. And then we asked, are those robust colonizers from sugarcane also robust colonizers for corn? And to do this, we sequenced the 16S from the microbe isolated from, the microbiome isolated from roots, stalks, and leaves of corn. And you see here, this is very important. This is a natural microbiome of the non-inoculated plant, and this is the microbiome of inoculated root, and then what happened in the stem and in the leaf. You see here that there is a kind of, the inoculated synthetic community displaced the natural microbiome. You see here, this bacteria here was gone, and the increase the colonization of the synthetic community. You can see here also an effect on the stem and in the leaf. And this goes from low to high. And then what we saw is that about 60% of the microbes that you find in the inoculated plants come from the synthetic community. So the synthetic community, this shows that the synthetic community, the robust colonizer of sugarcane, are also robust colonizers in corn. And then we asked, what are the features of encoded by those robust colonizers? Why? What is enriched using the same idea of gene enrichment? And what we found is that we sequenced the complete genome of all those bacteria from the synthetic community, and then we classified in enriched and depleted genes. And you can find here very, very, very interesting things. There is a huge enrichment of transporters in the genome of those bacteria. Transporters of what? Transporters of sugar, of organic acid, and some other compounds that represent the exudate that's produced by the plant. So perhaps those bacteria are robust colonizer because they can use very efficiently the metabolites that is produced by the plant. It's produced by sugarcane, and perhaps it's exudate in the roots, and then those bacteria can grow very fast and can overcome the other bacteria that is around. Okay? We found some other very interesting, what we call traits in those encoded by the genome, including some of the exopolysaccharide that is associated with colon sensing and protecting bacteria and so on. So we found also some antimicrobial bacteria, antimicrobial genes and things that you have shown also. But, well, then we asked, can this really impact plant growth and development and yield under stress condition? To do this, another student in the lab, Jader, constructed a fantastic phenotyping system, and this is an experiment that was conducted in a greenhouse. You can see here, he made sensors to measure leaf temperature, sap flow, soil moisture, plant weight, and also sensors to monitor the room temperature, light intensity, and humidity, and so and so. And then, when you see here, the pots in black are the pots that we monitored, and then we use 3 commercial varieties of corn, commercial hybrids from 3 different companies in this experiment. And you see here, and this is monitored, this is real-time monitoring. So we collect the information every 5 minutes along 4 months. So to do this, they collect 6 million data points, and then they integrate the data points. You can see here a window of about 30 to 85, this 55 days. And you can see here the temperature, the room temperature, sorry, I can't read here, but this is the humidity, and this is the photosynthetic active light, and this is the VPD. And you can measure this, you can follow, right? What happened? This is 1 hybrid inoculated in blue and this is another hybrid, non-inoculated in red. And this is the difference in temperature, in the leaf temperature between the inoculated and non-inoculated. And you see here that the inoculated plant, it keeps the leaf temperature more, let's say, constant, while the non-inoculated plant, you see here, there's a difference in temperature. So when the room temperature increases, the non-inoculated plant increases again. And you can see that this difference, there are peaks of the increased temperature of about 3 degrees. Remember that when we increase 1 degree or half degree in our body, we are starting fever. You can imagine what happened in a plant when the temperature increased 3 degrees in the leaves. Maybe you can have phenomenon of protein denaturation and things like that. And what is fantastic here is that the inoculated plant, keeps the temperature, so can help plants, maintain the temperature even if the temperature at the room temperature is higher, increase, right? Okay. This is another kind of measurement. You see here, there are many data here, but look here. This is the difference in sap flow between inoculated and non-inoculated. What is sap flow? Is we measure with sensors, the water movement from the pot to the air, all right? By putting in the stalk, putting sensors that can measure the sap flow. In blue, inoculated. In red, non-inoculated. And here, we introduced here an drought stress, so we just stop irrigation here, and you see that in blue, the flux of water is going up about the same in inoculated and non-inoculated, but then when the drought increase, you see here that the non-inoculated plant keeps sap flow continuously, and the inoculated plant stop a little bit the movement, the water movement from the pot to the top, all right? And then we irrigate again. When you irrigate again, you see here that the inoculated plant start, increasing the water movement. What does it mean? This means that the inoculated plant is able to maintain the temperature few degrees lower and stop taking up water and stop taking up water so that they can keep water in the plant. And this is, again, the difference in temperature with, during the stress and recovery. And here I'm going to show you a little bit about the behavior of the plant, because not only the sensor, we put also some cameras taking photographs every 5 minutes along all the experiment. And you can see here. Oops. See this? And then we re-water. This is inoculated, non-inoculated. Inoculated, non-inoculated. Inoculated, non-inoculated. You see here? This is a Monsanto, Syngenta, and Pioneer hybrid. What does it mean? That inoculation maybe helps plant to keep the turgor. And of course, this is a completely dry plant, so we just, stop irrigation and let the plants, to fall down, and just as a readout of the experiment. And this shows that the inoculation changed completely the physiology behavior of the plant. So there are something happens there that plant can keep the turgor, and this can maybe help plants to reestablish after irrigation quickly than non-inoculated plant. And one thing that is very important, there are differences between the hybrids from the different companies, because those differences may be associated with the genotype. The genotype of every hybrids are different, and there are some genotypes that respond better. For example, the Monsanto guy here inoculated, don't see, this kind of dramatic stress. So we are now trying to understand what are, what is the mechanism by which those inoculated plants, keeps the turgor and control the physiology, control the temperature, and control the flux water flow. And what does it mean in terms of drought tolerance? What is very fantastic in microbiome, because this can be a product already. We know now what are the microbes, what their genome encode. We know that we can play with a mixture. We can combine 1, 2, 3, or any kind of combination, because we can select this based on the markers that encoded by the genome of the plant. And this can now ready to be to go to the field. And we are already doing this in field trials and different places. Unfortunately, we put an experiment in a place that is in Bahia, that's called Luiz Eduardo Magalhães, that is very dry, but this year rained like we lose this information this year, but we are going to repeat the experiment, explore this in another place. So, and, oops. Okay. Now back to the campus rupestres. Why we went to the campus rupestre? Because we have an idea in the team that perhaps the wild environment can have, the diversity in the wild areas that has never been, faced with chemicals. Maybe there are the evolution, the association between the plants and microbiome in this area should be represent what is the real co-evolution of this association between bacteria and plants. So we went there, we select a bunch of plants, Velociacea, that are monocots, and then we sampled the soil and endophytic root, epiphytic leaf, exposed rocks, because there are some plants that grows in rocks. And we sampled also steams and so and so, and we did this again, sequencing the 16S and ITS, and this is the bacteria, or this is prokaryote, and this is eukaryote, represented by ITS, and there is a lot of variation and population or diversity and abundance in different samples can be used, to understand a little bit more about how the dynamic of microbiomes are important to keep plants growing in this very dry area, and this area also is very poor in phosphorus. So we are searching for new robust colonizers for phosphorus acquisition. Maybe we can find here, and then this can be applied to crops. So this is the work that we, one of the areas that we are doing in our center. There are many other areas that is being explored for drought resistance, including transgenesis and gene editing, gene wars, and so and so. And this is the group. We call this is Genomic for Climate Change Research Center. These are the groups. We have 28 people working in collaboration with different collaborators from academia and from also from Embrapa, and with just one focus: develop a plant that can grow with less water and perhaps less fertilizer and less pesticide, because we want to also to identify plants that can face, pathogens that are important for a plant. So thank you very much for your attention.
I'm amazed. And this change we made in the program was just complementary. So, wow, great stuff, great stuff. Do you take a break, or you take a break? Let's take a 15, 20 minutes break, and then we come back for the round table in 4. Yeah. Okay, let's take a break.